Next up, I would like to introduce Zachary Peters. Zachary is also a final year PhD at Dartmouth College in the United States. The title of his talk today is Immune Checkpoint VISTA on on keratinocytes, suppress Sting-mediated type I interferon responses that mediate skin injury by ultraviolet light. Zachary, feel free to take away.
Thank you so much for that introduction. Yeah, so I'm a PhD student in the Scopelio-Gardner lab at Dartmouth College, and I'll be talking today about immune checkpoint VISTA on keratinocytes and how it regulates those type I interferon responses to UV light. So our laboratory is really interested in the molecular mechanisms underlying systemic lupus erythematosus. And while we've already heard so much about the importance of these antiviral cytokines in the context of viral infections, they can also be pathogenically upregulated and persistent in many autoimmune diseases like lupus. So the focus of my research is really on cutaneous lupus disease. And so one of the key triggers of lesions in cutaneous lupus is exposure to UVB light. So UVB can really damage cells within the epidermis, like keratinocytes, and that can lead to the release of DNA into the cytosol and trigger interferons via the cGAS Sting pathway. And these interferons can then go on to other cells, bind to the receptors, and trigger expression of various interferon-stimulated genes. And both in the laboratory and in the clinic, we can measure expression of these genes and sum them into an interferon score that can give you a measure of the interferon signature within a particular tissue. So systemic lupus erythematosus is a very complex autoimmune disease, but the majority of these patients have some form of skin disease. And these patients have this elevated type I interferon signature, both systemically in their PBMCs, as well as their kidneys, and also locally in their lesional as well as non-lesional skin. So this interferon signature can present before a lesion actually even occurs. And importantly, these interferons are a real key mediator of the disease. So studies have shown that blocking the type I interferon receptor is probably one of the most effective ways of treating cutaneous symptoms. So patients that go on anifrolumab have this suppressed cutaneous lupus activity and severity index for up to 6 months. However, a lot of these type I interferon treatments don't address a potent trigger of lupus exposure to UV light. So previously, our lab has demonstrated that an acute dose of UV induces a robust interferon response in both healthy human and healthy mouse skin. And importantly, our lab has demonstrated that this early and local type I interferon response is really dependent upon that cGAS Sting signaling. So if you get rid of cGAS, you don't have this early interferon response. But this interferon response returns to normal over time in healthy human skin, or healthy mouse skin, you can see here. So we wondered what downregulates this interferon production, and could we find some target to inhibit interferon in lupus skin? So we believe that immune checkpoint VISTA is a suppressor of type I interferon responses triggered by the UV light. So VISTA is a negative immune checkpoint molecule among many other co-inhibitors that serve to regulate inflammatory responses. They've been predominantly studied in the context of cancer, where blocking these molecules allow T cells to go off and clear the tumors. However, agonistic antibodies also exist, where you can activate these checkpoints and suppress inflammatory responses, potentially in the context of autoimmunity. So we think VISTA is a key target to treat type I interferon for several reasons, including, for one, VISTA-deficient PDCs produce significantly higher levels of interferon alpha. And another lab at Dartmouth, Noel Lab, has demonstrated that when you stimulate human primary monocytes and treat them with an agonistic VISTA antibody, you suppress all of these interferon-stimulated genes compared with an isotype control. So perhaps VISTA is regulating either interferon signaling or interferon production. So to study whether VISTA is regulating interferon responses in the mouse skin, we performed bulk RNA sequencing on wild-type and VISTA-deficient Black 6 mice, young mice, and gene set enrichment analysis identified response to interferon beta as the top most enriched pathway in these mice. We validated this by performing qPCR for various interferon-stimulated genes, and the VISTA-deficient mouse skin had a 10-fold higher interferon score compared to the wild-type controls. Additionally, they had elevated levels of interferon kappa. So we next wondered what would happen if we exposed these mice to that acute dose of UV. So to do this, we shaved wild-type and VISTA-deficient mice, exposed them to an acute dose of UV, and from a subset of mice looked early on at their interferon response, and then others we looked 2 days later for their physical response to the UV. And so by performing qPCR for various ISGs, you can see here representative ISGs, and also by the interferon score that both wild-type and VISTA knockout mice mount a robust interferon response to the UV. But the VISTA-deficient mice have a much higher magnitude of response, about 5-fold higher to the UV. Again, 2 days later, these VISTA-deficient mice have this really severe inflammation and rash compared to the wild-type mice, suggesting that they have a lot more damage in their skin. So I mentioned early on that the early and local interferon response was really dependent upon that C-gas Sting signaling. And so we wanted to test whether this was really responsible for the elevated interferon and UV-induced skin injury in the VISTA knockout mice. And so we exposed VISTA Sting double knockout mice to that single dose of UV. And as you can see here, they don't experience the same amount of lesions and rash compared to the VISTA knockout mouse. And we can actually quantify that with an adaptive CLASI score here, and showing there's a reduced amount of CLASI in the VISTA Sting double knockout mice. Additionally, we've done some flow cytometry to characterize the immune cell infiltration in the skin 2 days after the UV. And as you can see, VISTA-deficient mice have this elevated level of CD8 T cell percentage, as well as a decrease in their T regulatory cell percentage. And the actual numbers of CD8s to FOXP Tregs is much higher in the absence of VISTA. However, these levels were all sort of restored in the VISTA Sting double knockout mice, suggesting that a lot of this is driven by Sting. Additionally, these VISTA knockout mice had elevated levels of Ly6C, we term inflammatory monocytes in their skin 2 days after the UV, which was also restored in the absence of Sting. So we put a lot of thought into what types of cells might be driving this worsened skin injury in the VISTA knockout mice. And I think probably the most important data we've collected over this time is to answer this question of whether adoptive transfer of VISTA-deficient bone marrow could induce skin injury in the wild-type mice. And so to do this, set up these bone marrow chimeras, we transferred VISTA-deficient or wild-type bone marrow into wild-type or VISTA knockout mice that were irradiated. We let them rest for about 8 weeks prior to an acute exposure of UVB. And here are the regular wild-type and VISTA knockout mice. You can see those lesions in the absence of VISTA. And the wild-type mice that received the VISTA knockout bone marrow, surprisingly, did not have the lesions that we see in the absence of VISTA. However, conversely, the VISTA-deficient mice that received the wild-type bone marrow had pretty severe lesions. And so when we look at the CLASI score, the VISTA knockout as well as the VISTA knockout recipients had really severe, really enhanced CLASI scores. But transferring the VISTA knockout immune cells into the wild-type mice did not induce that skin photosensitivity. So this really suggested to us that the non-immune cells might be playing a role in this skin, the enhanced UV-induced skin injury. So when we think about non-immune cells in the skin, our first thought is to the keratinocytes. So they're the majority cell type within the epidermis that's getting damaged by the UV. Additionally, they secrete a type 1 interferon called interferon kappa, which is one of 2 type 1 interferons enriched in the skin of lupus patients. And so first, we wondered if keratinocytes actually express VISTA. And so we've demonstrated a number of ways that VISTA is on the surface of these cells. So here is an immunofluorescence image of mouse epidermis. And you can see co-localization of VISTA on with cytokeratin 14, an epidermal keratinocyte marker. Additionally, we've shown that both primary as well as human keratinocyte cell lines express VISTA. And you can see by this Western blot that exposure to UV can actually induce VISTA expression alongside interferon kappa. So to study a role for VISTA intrinsically on keratinocytes, we use these mice with a conditional deletion of VISTA in their keratinocytes, these VISTA-flocked KRT14 Cre positive mice. We performed bulk RNA sequencing on the skin of these mice, and like the total knockout, response to interferon beta was the topmost enriched pathway in these mice. We validated this with a qPCR for various ISGs, and these mice, again, like the total knockout, had about a 10-fold higher interferon score compared to the VISTA-flox controls. So this was really surprising to us that just loss of keratinocyte VISTA could have this effect. Additionally, we've characterized a number of interferon levels, and it seems like interferon beta is really enriched in the skin of these mice when you lose keratinocyte VISTA. So we next asked what would happen if we expose these mice to that acute UV. And the mice lacking keratinocyte VISTA really had this severe rash, similar to the total knockout. So when we look at the CLASI scores, you do see that enhanced CLASI in the absence of keratinocyte VISTA. So importantly, we needed to understand if this enhanced skin injury was dependent upon type I interferon. And so we treated our VISTA-flox and KRT Cre mice with an anti-IFNAR antibody or isotype control 3 hours after the UV exposure. And the KRT Cre mice that received the anti-IFNAR blockade had reduced lesions compared to the ones that got the isotype control. So blockade of IFNAR reduced the CLASI scores in the mice that were absent of keratinocyte VISTA. So just to conclude, I think we've demonstrated that VISTA is a suppressor of baseline and UV-induced skin interferon responses. And a lot of these interferon responses are driven by Sting. And additionally, it seems to play this role on keratinocytes in suppressing both the UV-induced interferon as well as that enhanced UV-induced skin injury. So I just want to acknowledge my lab for all the teamwork and support they put into this project, as well as my PI, Slajana, for her mentorship for this project. And I'll conclude there.